this is a lecture about the phasor approach, application. Okay, so let me see if I can move up and down. Okay, so I want to start with some sort of background in lifetime, although this is not needed after Dave's lecture. And the introduction, I want to talk about what is lifetime imaging microscopy, which was the goal of the first grant I applied, and then what is the motivation for FLIM and where it comes the phasor approach, and then I will talk about some biosensors. Okay, so let us see the next. Okay, so as Dave explained, a system, an electronic system is always in the ground state S0, and you provide energy and you can excite the state S1 or S2 or S3. And if you have only one state, then the system will decay with an exponential, as is shown here in this graph. Okay, so the going from whatever excited state S1 to a bottom of S1, it takes about 1 picosecond per vibration. And this is because a vibration takes about a picosecond to occur. And then what you measure is the emitted fluorescence, which is shown in red in this figure. And now when you do images, you need to collect images in the decay curve. So for example, that one will be after a few nanoseconds, that will be after a few nanoseconds, this will be in nanoseconds. And you can see that every image will contain on the order of 1 million points if you have 124 by 124 images. And then the decay of this image, so if you analyze the decay in the image, in each pixel in the image, you can obtain the decay of the fluorescence in that. Okay, and Dave explained that for those like us coming from the Gregory Weber school, instead of measuring the decay, as is done by people working in single photon counting, we measure the phase of the sample. We expect to say of a reference. And that is the idea that there is a delay. For example, the maximum of this curve and the maximum of the other curve is different, and also the mean. So FLIM is used, for example, for PET. I will talk a little bit about intracellular mapping of ion concentration in pH imaging, biochemical reaction process like NAD and NADH, which is probably the most popular application of FLIM, and long lifetime imaging for phosphorescence, for example, for the determination of oxygen concentration in cell and in tissue. Okay, so let us start with the PhET. So PhET was developed in the quantum mechanical point of view by Theodore Foster, and the paper is 1949. And in the description of the process, you have a donor which gets excited, and the donor can transfer the energy of the acceptor if some conditions are met, and then you end up with a system in which the donor is in the ground state and the acceptor is in the excited state. And this is in terms of a Jablonski diagram, what happens in this system. Okay, so in order for that to happen, you need some condition, as was said by Dave. So first of all, you need a rate of energy transfer. And this rate of energy transfer goes with the 6th power of a quantity, which is called R0, which is measured in angstrom, and depends also on the lifetime of the excited state. So R0 is a complex quantity, which depends on the overlap integral between the emission and the absorption, depends on the kappa square, so it's the angle between the absorption and the emission dipole, and then depends on the index of refraction, and depends also on the quantum on the quantum yield of the donor. And this is the formula that Foster produced in 1949, and this is essentially accepted by everyone. There are different kinds of energy transfer. I will not talk about energy transfer due to other processes like electron transfer and so on. But electron transfer gives information about distance between two groups, because you can measure F0, orientation between two groups on the kappa square, and notice that in principle the donor and the acceptor can be the same, because then in that case there will be overlap, and so in that case it's called a homotransfer. So because it was about a similar thing. So if we plot the energy of transfer as a function of the distance in angstrom, we get the plot as shown in the figure here, and this is the plot that says that the efficiency of energy transfer can be determined if you know the plot. So, for example, if you know one plot of another point in the plot, you can solve for the energy transfer. And so the energy transfer energy is defined as the decay due to every kind of process that will produce a decay of the excited state divided the sum of all the other state. Experimentally, what we measure is a quantity which is very simple to measure in lifetime, which is the quantity that has to do with the lifetime of the donor in the presence of the acceptor and lifetime of the donor alone. And those are samples which are easy to produce, because, for example, using fluorescent proteins, you can have a sample which is only have a donor and acceptor, and the sample that only have the donor only, and then you can calculate the efficiency of energy transfer for this formula in lifetime. You can calculate also from the change in energy, which was done before lifetime was available, and you can obtain the same result. And you can see that, for example, you can transfer from D to D to A, and so on. So you can transfer, provided there is an overlap between the emission of the D and the emission of the D and then etc. You can change, you can move the energy transfer from the value you want. So this is a plot of the chameleon, which was one of the very first scan sensor produced by Roger Sien, and you can see that you have a emission of CFP and the emission in yellow of the YFP, and then by measuring the intensity, you can measure the amount of energy transfer. This is what Roger Sien did, and it works very well. And then he said, no matter what you do, you have a ratio, which is how much will the decrease the intensity of the donor to respect to the acceptor, which will be the other peak. And I give this talk or similar talk in his lab, and he said, well, no matter what you do, you cannot improve on what is there. Okay. Well, you will see that actually you can improve, because you can measure only one element or only the other element, and actually that improves the capability to see the energy transfer by very much, no matter how small it is. Okay, so let us talk a little bit about conceptual approach in spectroscopy. So if you have, for example, spectrum of CFP and YFP that are mixed, you can measure the lifetime component, and if one lifetime component, for example, the first one is goes with FFP, and the longer one goes with the YFP, you can demix the system and say how much CFP and how much YFP you have. In order to do that, you need to know the spectrum. So you need to know the spectrum of CFP and YFP. And this is generally not known a priori. And the same thing happens for also spectral phasor. So you can do a spectral demixing, but you will need to know the components of the emission. And this is what most of the software do. Okay, so for example, for a red pair, you will need to measure the fluorescence of the donor only, the fluorescence of the acceptor only, and then map where that happens. But let us talk about the challenge of FLIM. Why it took so long to develop a FLIM? At every pixel in an image, you have a contribution of several fluorescence which each one could be multi-exponential. So you need to know those components if you want to solve a pixel, the lifetime of a pixel, which is not in the cell. To make things work, and this is the warning, you can only collect light for a limited amount of time, let us say 100 to 200 microseconds per pixel. The reason for that is that if you want to measure 1 million pixels, clearly when you have 1 million to that, you will get a very different thing. So this is really the maximum amount per pixel that you can have, which result in about 500 to 1,000 photons per pixel. Now, if you simulated the cake with 500 to 1,000 photons per pixel, this is not enough to distinguish a double exponential from a single exponential. And this is important because people are trying to resolve a double exponential or whatever, and they cannot distinguish a double exponential by a single exponential, simply because the fit will not work. By solving the decay pixel in multiple components involves fitting a function. And this is traditionally a complex computational task, and we say for expert only. Okay, and this is a problem because I don't like the fact that only an expert can do this analysis, and I would like to have something that can be done by everybody. So it is a rather difficult technique because you need many photons. Fitting is low. Generally, a fitting of every point of an image using different minimization method requires sometimes in the order of 1 hour. And I don't know what is for you, but if you have to wait in front of the computer for 1 hour, your mind goes on something else. Result, the most important thing, the result depends on the initial condition. So if you are an expert and you know what the initial conditions are, well, you can do better than if you are not an expert because you have to try everything. So the question is, now I was in this condition, and then the condition is essentially, can we avoid all those problems? So, for example, no expertise is necessary. And this is very important because that means that everybody in just in no time can become an expert if no expertise is necessary. Can we have instantaneous results? So instead of waiting for the minimization to occur, can we get instantaneous result? Can be independent of the initial choice? And can we have quantitative result and a simple intuitive interface? Well, this is really a very important question. And in order to do that, we need to change a paradigm. So the paradigm will change if we propose to have a different representation of the decay, where each molecular species has its own unique representation, where each process, Fred ion concentration changes, is easily identified. So I want to show you how that is. And that is not done by simply having better program for minimization and so on, but it's going by done to a new space. So what is this new space? This new space is a space, which similar to Star Trek. And then we have a gun that is powerful enough to reach everyone in every place. And if we transform the data and we go in a different space, then we can have a very simple representation of the decay. So each component, each decay will have one point only instead of many exponential decay and exponential factor. And where is this concept coming from? So this is where we started. So we have a different point in every image. And so we have to resolve the system of the decay of one component in different points. So the interval between excitation can be between, that curve can last a nanosecond or two. And then you need to acquire the data for the entire image in a nanosecond or two. And then let us see what is happening. Okay, so what is happening is the following thing. If we follow what Gregorio Weber was able to do in the 70s, he said, well, if we know the decay of the intensity, which is here, so if we measure the decay of the intensity, we can define two coordinates, the G and S. So the G is the cosine transform of the decay, and S is a sine transform of the decay. So if we do that, so instead of having a decay, which is very complicated, we can be the sum of many components, we have only a G and an S curve. And so for every decay, at that point, we will have a G and S. And G and S are coordinates of a point. So the time domain, in the time domain, so we start from the time domain, and we have a component of a phasor, which are G and S. And IT is the measured one. So let us see how it works. So it's not only just the Fourier transform, it's the following thing. So first of all the points will have a modulation and the phase. The phase will be larger. The larger is the lifetime, which is measured along the universal circle. And that in this axis we plot G, and this axis we plot S. So we have a universal circle which determines the points that are inside the universal circle and points which are outside the universal circle. And we will have example of both, and you have seen from Lionel a presentation that points can be outside or can be inside. So the important thing is that the single exponential lifetime must be on the universal circle. So if points are on the universal circle, and then you have a single exponential decay. If you have multi-exponential decay, we can see, so suppose this is a one exponential decay, and that will be another exponential decay. All the points will have two exponential decay will fall in the line of T1, T2. And so an experimental point will be here, and the distance of this point to T2 and the distance from this point to T1 will give the fractional component, fractional intensity component of T1 and T2. And we'll give the fractional intensity component because we divide by the total intensity. Okay, so now we have the exponential decay, and instead of measuring every point, we can do the fitting of exponential that is done in the time domain. We can, for example, if we do something like that, we get a long lifetime, which will be that part of the curve, and the short lifetime. And people, and there are many papers in which the long lifetime is considered to be the free die, and the short lifetime is considered to be the exponential decay. This is not true, because as I show, the short lifetime can be the linear combination of two components. So let us see how we can separate different component lifetime. This is an easy experiment. And I have 3 capillaries, one filled up with fluorescence, the other with rhodamine B1, and one which is the mixture of the 2. So we have in the phasor plot the points corresponding to that capillary, the point corresponding to central capillary, and the point corresponding to the D. And we have 3 components. So L, F, D. And the 3 components must lie in a line because fluorescence is a single exponential, rhodamine B1 pure is a single exponential, and we have an intermediate point, which is the combination of the 2. In order to determine the component that we have in TEF, what we do is we need to calculate the distance between this point and the other point, and this point and the other point. And if we have that, we have the component. And this is, we have a program that is used in order to do that. And the program, the result are here, for example. You can see here that what is the left line that you can measure in phase and modulation. So now suppose we have a different sample, not something we know everything. And this is a sample of a cell growing in a matrix that contains all the molecules. And then you can see that from this point, which is the highlight by taking all the points in the cell to another point which is close to 0, you get a linear combination action. If you pay attention to that, you will see that if you select the points which are in the collagen here in the linear combination, you get those points. So you have one point and the other point. And the relationship between that point and the other point will depend on the linear combination of the 2 components. So that means that if we have a sample like that, we can distinguish immediately the point we have one component, which would be that one, and the point we have another component that will be the other one. And then the points which are a combination of whatever is the background and the linear combination that will work only on the body of the cell, because those are the points in which we have some EGFP and some of the background. And you can see that is easy to distinguish, and that will show you the things in a different way. And then you will see that one, which is the cell labeled with the GFP, 2, which is the background, and 3, and you can see that 1, 2, and 3 are points that you can obtain directly from the measurement. You don't need to do anything. And this plot can be obtained by anybody. And you will have 1, 2, and 3 components. And you see that you have 3 linear combination. You have a triangle, so 1 and 2. So will be all the linear combination in which you have a little bit of 1 and 2. 2 will be the collagen, and 3 will be background fluorescence, because everything after you have also background fluorescence. So you can see that without any effort from outside, we can determine exactly the components of the system. So we have, in this system, we have 1, 2, and 3 components. And there is no doubt, everybody with the experience or not will reach the same conclusion. So if you have more than 3 components I was showing, so for example, the red and the green, here, the red and the green, you have a linear combination. But every point of the linear combination will combine in order to give another point like the blue point. So in the case you have 3 components, you have 1, 2, 3. If you have 4 components, you will have 1, 2, 3, 4, and so on. So it is clear that the components, if you have a pure component, a part of the sample has a pure component, but a part of the sample that have another pure component, you can you can immediately distinguish them and analyze them. So this is a picture of a paper, of the paper we published with Michel Digman and others. And for example, that was this point of YFP. CFP clearly are very similar. EGFP is in a different position. We have the cell autofluorescence, which is more or less in the center of the plot, and the fibronectin, which can be in another position. And then we have here a line which indicate a biosensor, RAICHU, RAC1, and these different values, and I will show you why it's different values. Okay, so how do we distinguish multi-exponential from FLIM? So in the multi-exponential point, there is a linear combination of a pure dye and a pure dye, and will be in any point of the experimental point, and then experimental point should be in the center. This point in the center cannot be anything but the combination of P1 and P2. And if you have threat, what will happen is that the threat will decrease the lifetime. In some way, it will go always in an orbit which is like that. This orbit is very difficult to calculate, but the computer will do that for you. So you say to the computer, given P1, P2, where will be the quenching trajectory due to threat? And the computer will plot on the line. And as we will see, so there are tools that you can use in order to quantify better. One is the fractional intensity calculator, and the other is the threat. So in the threat, generally you have a donor that will be quenched, so it will move in that direction, so short lifetime. An acceptor that, for reason that we'll tell later on, will move outside the universal circle, because the acceptor is excited after the donor, so it's on delay, and will move outside. And so the calculator will follow the following thing. So a donor which is quenched by transferring energy to an acceptor will be on the universal circle, because if the donor is a single exponential, will follow a single exponential too. Now, generally, the donor is not a single exponential, and then you will have many points that bring up to the 1, 1, which is a zero lifetime. And then you will have a curve which is difficult to calculate by hand, but the computer will do it exactly. Okay, so the threat calculator will calculate. So you have a donor, and then you have 10% of acceptor, etc. And depending on the amount that you have, will end up, a curve will bend, and it will end up in the autofluorescence, because at the end, when you quench all the donor due to transfer or not, you will have only the fluorescence. So the donor and the acceptor have 2 points of the phasor diagram, and you can immediately, by analyzing only the part of the curve, you can tell how many autofluorescence you have in the presence of the donor. And then all the points will be in this side of the thing. So this is an example, and then you can see that, for example, starting from a donor, which is here, the donor will follow a complex trajectory. The experimental point are here, and for experimental point, I will calculate the threat efficiency, which is 0.292, and then we'll calculate the background autofluorescence and the molecules which are not quenched at all. And then you can see that the threat calculator will draw that curve for you. You have simply to say where are the points, and then the points will, if you move out or in this point, you will change the score, so the number of points that are contained in that thing, and then you can maximize this value just by moving the cursor there, And by maximizing this value, you will determine exactly how much molecules perform FRET and give you the FRET efficiency and all the other parameters that you need for the analysis. Okay, so now let's go to a system which was in the paper we published with Michel Dickman and others. And the system has cells. Let us see if you can distinguish the cells here, and so on. And if you look at the intensity, very few points have more than 1,000 pixels per, probably those 2 points into the point. And the rest is really below 100. And from here to here, you cannot distinguish the word exponential. And this is a system that has the following problem. So we have a cell which is expressing the UPAR receptor, and the UPAR has two, can be expressed with EGFP or MFR. So, and then we measure A, B, C, and D points, and let us see what happens. So, in every point, we will obtain a value of whatever exponential they are. But if we look at the entire cell, we get this cell which contains, I will tell you what is then, but this is this peak, and the other cell which is less, so much less than, which is the other one. And now let us see if it's possible that this peak is due to FRET or not. Of course, you already know that it's not so. If we select all the points which are in this part of the histogram and we show them, we have that cell. So this is the blue line indicate the one dye, the other dye, and you see that they are in linear combination. And if you want to get the dye in this part of the cell, there is even less linear combination. So it will be far. But the FRET in this system can occur only at the junction between cells. So the junction has to be like that. Now, if you pay attention, hello? If you pay attention, you will get some points which are in this part. And then the computer will show you all the curve for different FRET efficiency. And so the FRET efficiency shows essentially how many of the molecules have YFP and CFP, one close to the other. And this is a sensor for FRET, for measuring the FRET. And so we can do, for example, different kind of sensor. For example, this is GT1 CFP, and you can see that this expresses everything in the cell. And the left hand of the background is in a different position. And then you start to see the development of the curve that will go to zero. It will not go up to zero because, of course, that means that every donor will have so this is the final curve, and you will get that you start from here and you start from here. So you already know those points will have some sensor and no sensor. And then the curve, you see, always deviate from the linear combination, and it goes in the area of, and this is a study that Michel did in using, for example, how many points are there, so that is low FRET, high FRET, and so on. And then you can say what are the point which you will get a very high FRET efficiency. And you can analyze an entire system just by moving cursors, as we did here, and figure out how much FRET efficiency do you have for this sensor. And this is a very, very simple thing. And every student in the lab was able to do calculate how many points you have here, how many points you have here, and here it says you the thread efficiency. What is the thread efficiency? For example, for this morning will be 42%, and that is the entire trajectory. And remember, the entire trajectory is done by the computer, so you don't have to do it. So this is a 8% thread efficiency and so on. So with this particular biosensor, it shows points that have thread efficiency very high, very medium, very medium, very high, and very low. Okay, so let us see if we can figure it out some sort of conclusion. Many of the obstacles of filters can be removed using this conversion to phasor and to frequency domain. The accuracy of the determination is improved because we can determine exactly where a point it is. The speed of the analysis is reduced in almost instantaneous for the entire images of several images. You can load several images and analyze all of them simultaneously. And this is very important because, for example, in the ratio, you can see which cell will produce more thread and less thread. The interpretation of the FLIM experiment is for a minimum prior spectroscopy knowledge is needed, which is what we want to have. The phasor analysis method can be applied to all modes of data acquisition, frequency domain, and time domain. And so you don't need to be an expert anymore because the data are plotted in a way that you can see immediately where they are. And the analysis of the trajectory of phasor plot provide the quantitative resolution of process. So you don't need to be an expert and so on. So I hope that I convince you that transforming the data in the phasor analysis, everything that seems to be complicated, very complicated in the analysis of the decay of an entire image can be done. Yes, can be done simultaneously for the entire image, and I will finish it.